Welcome back. Today we are talking about important eukaryotic cellular structures and their functions. I'm going to divide these structures into two different systems. One is the endomembrane system of organelles that I've listed on one side, including the nuclear membrane, smooth and rough ER, Golgi, lysosomes, vacuoles, and plasma membrane. All of these either structures or organelles, we'll talk through that. And then some outsiders like your energy organelles like chloroplasts and mitochondria, and a few other organelles, cell wall, cytoskeleton, cilia, and flagella, that make up some of the structures that we'll be talking about this particular unit. So let's take a look at some of these structures and note their function. We're going to start with the endomembrane system, and I'm going to start with the nucleus, so that internal structure, and sort of work from the nucleus all the way out to sort of that plasma membrane. So working from the inside of the cell to the outside. Now, in order to belong to the endomembrane system, it has its own kind of evolutionary origin of folding of membranes, that external membrane that we talked through in our evolution unit. But basically, to belong to this system, you have to be connected to this system of membranes that extend all the way from the nucleus all the way to the outside surface of that plasma membrane. Now, you can be related and connected into this membrane system by direct contact. So sometimes membranes will lay right next to each other and receive information, or through membrane-bound containers called vesicles. So these are little membrane pouches that hold different materials or signals that go from one of these organelles to the other. So either you are connected to the system by direct contact or by these vesicles that are transporting materials around the cell. Either way will connect you to this endomembrane system. The endomembrane system of organelles has lots of different functions, everything from creating and packaging proteins to dealing with lipids and metabolism of different kinds of sugars, fats, and then also breaking things down that are necessary for the body and building different materials that are necessary. So lots of different functions for this particular set of organelles. So starting at the center of a cell, the nucleus, some groups of textbooks include the full nucleus, some include only the nuclear membrane. But it is true that the nuclear membrane, you can see on this picture, lays right next to the rough ER and the endoplasmic reticulums that lay just outside of the nucleus. The nucleus is going to be sending messages to those rough ER, those ribosomes on the rough ER and free ribosomes to build certain proteins. And so the two are greatly connected. The overall function of the nucleus is to house and protect your DNA. So DNA in a prokaryote is just loose, but in a eukaryote, it's protected in this membrane-bound container. That container down here, you can see these rosette holes. These are nuclear pores. These pores then allow their own kind of transport. So they'll block certain materials from entering the nucleus and touching and interacting with DNA and let others come in and out. And so those nuclear pores allow for certain materials to sort of be distributed outside of the nucleus and stay out so that they're not like sort of touching and harming DNA. Lastly, your nucleus, besides protecting and housing your genetic material, it also houses this, usually it's seen as a dark spot in a nucleus. It's called the nucleolus. And this nucleolus is the structure inside the nucleus that helps create these ribosomes. And ribosomes are not an organelle, but they are a structure that can be free or attached to the rough ER, and they are the structures that help build proteins. DNA, remember, codes for your protein traits. So it makes sense that DNA is going to talk to these ribosomes, and ribosomes become almost the workbenches where we build the proteins based on what DNA is signaling out to these ribosomes to build for any given day. Your nucleus then is almost like the control center, right? It's almost like the director of an orchestra. There's all these different things going on in the cell, and the nucleus is going to be sending out signals of when to sort of play and when to sort of create things, when to break things down, and how to sort of work the general workings of the cell. So it's going to be the main sort of director of the cell. If we go from the nucleus, just on the outside of the nucleus, typically we encounter the rough ER and the smooth ER. These are known as the endoplasmic reticulums, and they are a highly folded set of membranes. You can see them in an electron microscope here and then kind of drawn as well. So they're highly folded. The difference between the two types of ERs, rough ER is embedded with ribosomes. So its overall function is to help make proteins because that's what ribosomes do. Smooth ER lacks those ribosomes, and so it's really a lipid set of membranes. And so really its function is going to be about processing fats and lipids and anything related to that class. So that includes steroids and hormones and some of those groups as well. These two kinds of ERs and folded membranes are almost like the factory lines in a factory, right? It's where you're going to build your main products, right? Whether they're proteins, but also these fat and nonpolar classes of chemicals as well. And so these are almost like the factory lines inside of the cell. If we move out, usually the next thing that you might encounter is the Golgi apparatus. And the Golgi apparatus almost looks like fish bones. I think it looks like a fish with almost like these sort of bones that you can see a skeleton inside your cells. This particular organelle is all about packaging proteins. So once a protein has been made on the rough ER or on a free ribosome, it's going to be received or often sent to the Golgi apparatus. It will be received by the Golgi, and then the Golgi is going to finalize that protein. Finalizing or packaging a protein means folding sometimes the protein into its final shape. So sometimes a protein is built and it's a long, maybe pearl necklace, but you want it to be a circle. And so the Golgi will fold that set of, pearl necklaces or those amino acids into like a circular shape or a sheet or a pleated sheet or a helices, lots of different shapes. So one of the packaging features might be folding the protein into its final shape. Sometimes cofactor and tags are added. So like different kinds of metals or different kinds of vitamins, minerals, these kinds of things need to be added, right, to a protein structure. These might be things that you're eating, but to finalize a protein, they need to be embedded into that protein in order to make it work. Think about like hemoglobin. It has this loop of proteins, and then embedded in the center is this iron complex that gets placed there. And lastly, sometimes proteins, we wait to turn them on until the last second. So they'll go to the Golgi, and then their shape, some kind of chemical reaction will have on the external shape or internal shape to turn it on, because we don't want proteins working sometimes before we're ready for them. Imagine enzymes that can break down different parts of your foods and things like that. You don't want them working in your stomach unless you have food around, and it's necessary to have them work, right? So it's a way to kind of save them and then turn them on at that last second. Lastly, the Golgi apparatus might package the protein with some kind of external signal so that it is ready to be shipped to the correct place. Some things some proteins will stay inside the cell, but some proteins, like in the human body, might be made in the cell and then transported to the blood or the brain so that it can have some kind of impact. So lots of different ways that we do that. If we go out, there's some external things that we often see. Lysosomes are one of these. Lysosomes are only in animal cells, so we don't see these in plant cells. Lysosomes are kind of like the trash can of the cell is my analogy, or the waste bin in the cell. They can digest food. They can digest damaged organelles like a mitochondria that's worn out so that you can replace it with something that is working. Sometimes in immune system cells, they will digest different kinds of viruses or bacteria. And my favorite thing that lysosomes do, if you ever think about like babies and how in like early development, how they go from a ball of cells, right, into like fingers, right, like digits and things like that. Well, lysosomes can actually open up their contents and dissolve away a cell. It's called cell programmed death or apoptosis. And what happens is these lysosomes can actually eat their way from this ball and eat out the different like digits so that you get this different form. You go from a ball of cells into sort of the final form of like the different presentation and the structural form of an organism. So that's pretty interesting. And I show that down here. You can see these lysosomes eating away the space between the digits so that you have like a 5-fingered baby, right? Or something like that. Another structure is a vacuole. A vacuole is for storage. So it's like the company safe. This can store things like sugars or water and store things from later. Some kinds of vacuoles are contractile vacuoles. That means they're like pumps. So they'll store things, but also like maybe pump them in other places, either out of the cell or into the cell, these kinds of things. For plant cells, vacuoles are very, very large. You can see this central area and how much of it is taken up by a vacuole. In an animal cell, we still have vacuoles. They're just smaller, and so they're not quite as big. The last structure that we hit, it's not an organelle because it's not a membrane-bound container, but the last structure is the plasma membrane. The plasma membrane does interact with all these other endomembrane system organelles, typically by vesicle transport, either directly or indirectly. And so I'm putting it on the list. And the plasma membrane, remember, its function is to really regulate what enters and leaves the cell, hold the shape of the cell, and really separate the internal contents from the external contents of the cell. It's almost like the gatekeeper, right, at the edge of a castle that's allowing certain things in and out of the castle community. So that's the end of the endomembrane system. Remember, to belong to that, you need to either touch or have vesicle interactions, right, with that particular membrane. There are some outsiders, so some organelles and structures that are outside of this membrane system. They don't directly touch the endomembrane set of folded membranes, or by vesicle transport, they don't like interact in the same way. One of those is the chloroplast. The chloroplast, its job is to capture sunlight energy and build a simple sugar. Now, lots of people come to college and they think that plants only have chloroplasts and animals only have mitochondria, and that is not true. Many plant types of cells are going to have both, right? And the reason why is because a sugar is a stored form of energy. It is not kind of the spendable form of energy that a cell is going to use on its daily, sort of day-to-day life basis. And so my analogy is that a chloroplast is almost like you're storing Canadian dollars in a US bank account. If you were to take your Canadian dollars, they are money, but if you were to take them to a store in Moscow, Idaho, then the store would probably be like, nope, we can't take those, right? Only US dollars. And so it is a kind of money, but it isn't a spendable form, a quick spendable form of money for a cell. So most eukaryotic cells will have a mitochondria as well. Not all, because there are some other energy pathways, but a lot of them will have a mitochondria, because a mitochondria allows us to convert these sugars and food, these stored forms of energy, into adenosine triphosphate, or ATP, which is really the spendable kind of money, the easy spendable form of energy, so that the cell can do its life work. So a mitochondria, my analogy is that it's like converting Canadian dollars. It's like a bank converting these different currencies like Canadian dollars into US currency, into this ATP form, so that the cell has money, right, or energy to spend to do its life work. Some other outsiders include the cytoskeleton. Its functions include to give shape, so it can be almost like the girders inside the cell holding it open. Some of these kinds of cytoskeleton can grow to the outside surface and become movement structures like flagella, which is like a tail-like structure, or cilia, which are like hair-like oar-like structures that move cells around. The cytoskeleton also participates in cell division by attaching to chromosomes and moving them around. And the cytoskeleton can also anchor the nucleus. The reason why the nucleus kind of stays in the center, typically, of the cell is because it's attached to cytoskeleton that kind of anchors those particular organelles in place. So this is like the beams and the walls of the factory that holds it all together. And finally, the cell wall is an external structure to the cell membrane. So you can see in this diagram, you get the plasma membrane on the inside space, and then a cell wall on the outside. Cell walls are not in every kind of cell, so some bacteria cells have them, plant cells have them, fungal cells have them, animal cells like our own cells do not have them, right? The purpose of a cell wall, it gives and provides shape for a cell. It helps filter some materials into and out of the cell, and also helps with water balance. We talked about how plant cells like to be turgid, right? And so it's almost like a balloon in a bottle. box and it saves them from lysis. So my analogy for this structure is it's kind of like the outside skin, right, of the cell around that plasma membrane. So these are the main structures of a cell, and just reviewing what some of their functions are. See you next time.